Welcome to the video on difference between discriminatory dissolution and QC dissolution or quality control dissolution. This is a very short video, and in this video you will be able to understand the exact differences between these two types of dissolutions, their purpose, their use, method of design, and role in the regulatory. So starting with the discriminatory dissolution, always understand that discriminatory dissolution is mainly done in the development phase of the formulation, and always discriminatory dissolution testing is performed with a dissolution profile. So multiple dissolution time points are selected, and for that the dissolution profiling is done. The purpose of discriminatory dissolution testing is to differentiate or distinguish between the formulations with suitable differences or meaningful changes done to the formulation composition, manufacturing procedure that may have impact on the drug's in vivo performance or bioequivalence and bioavailability. Discriminatory dissolution is to identify difference in the formulation that could affect the bioavailability, and discriminatory dissolution testing is performed in formulation development phase. During the development, it is done to detect variation in the formulation or manufacturing processes and differences in the particle size like extreme types, manufacturing methods, variations in the CMAs, CPPs, and the stability conditions or the packs. Method of design for the discriminatory dissolution testing is to be more sensitive and stringent than the QC dissolution. QC dissolution is a straightforward type of method, and generally single point dissolution testing is performed. This might be used for specific conditions like change in the different media, pH levels, volume, apparatus, and agitation speeds. These changes can highlight the differences in the formulation's release characteristics so that the formulations can be differentiated. It can be used to support the formulation changes that are important in establishing the bioequivalence correlations, that is IVIVC, in vitro in vivo correlation. For this purpose, discriminatory dissolution testing and dissolution profile is done. Also, you can justify the formulation differences based on to the results of discriminatory dissolution profile. Coming to the QC release profile or quality control dissolution testing, this is generally done on single point or Q points. Purpose is to ensure consistency and uniformity between the production batches and to release the formulation batch into the market. This QC dissolution testing verifies that the manufacturing process of the product consistently produces a product that meets the predetermined quality specifications. And QC testing is routinely used as a part of routine batch release during the manufacturing phase or commercial manufacturing phase to confirm that every batch of a product meets its established release criteria or finished product dissolution specification before it is released into the market. And QC release dissolution testing results are included in the COA of the formulation batch. Method of design. So QC dissolution methods are typically simpler and standardized. These are used as plant levels in the commercial setting, using the conditions that are consistent with what is specified in the drug's official compendium like USP, IP, and QC dissolution testing is approved by the regulatory authority. While discriminatory dissolution method is only in the formulation development phase, so it is not required the approval. The goal is to ensure that the product meets the defined acceptance criteria rather than to identify the differences between the formulations. Regulatory role of QC dissolution method. QC methods are critical for regulatory submissions and batch release, ensuring compliance with the approved product specifications. So the Q point, the dissolution parameters, and the specifications of the QC dissolution testing are approved by the regulatory authorities to release the manufactured batch into the market. So this is regarding the quality control dissolution testing and discriminatory dissolution testing. Now we will see the differences. Discriminatory dissolution is to distinguish between the formulations with suitable differences that may affect in vivo performance, that is bioavailability and bioequivalence. While QC testing is to ensure consistency and uniformity between the production batches before the product is released into the market. Discriminatory dissolution is primarily used during the development phase to refine and optimize the formulations. So this is to optimize the formulations, to select the suitable composition, to finalize the CMAs and CPPs. While QC testing is used as a part of routine batch release during the manufacturing phase. So discriminatory dissolution is used in the formulation development phase, while QC testing is used in the manufacturing phase. Discriminatory dissolution testing is designed to be sensitive and stringent, may use different pH levels, media types, and agitation speeds to detect the differences between the formulation batches. And QC testing is typically simpler and standardized, following conditions like monograph conditions and compendial guidelines, USP, EP, IP. Discriminatory dissolution method focused on identifying variations due to changes in formulation manufacturing process like CPPs, CMAs, and stability conditions, packaging materials, particle size, excipients, manufacturing process variations, etc. While QC dissolution method and testing is focused on ensuring batch-to-batch consistency to meet the predefined acceptance criteria of the finished product testing. Discriminatory dissolution is used to support formulation changes and to establish in vitro in vivo correlation, that is IVIVC. Also, this method can be used to have biowaivers, while QC testing is critical for regulatory submissions and compliance with approved product specifications for batch release. So for QC testing, the specifications are required to be approved, and QC testing is to match or meet the acceptance criteria for batch release into the market. Discriminatory dissolution is more sensitive to differences in formulation, helping to optimize and ensure robustness during development. While QC testing is somewhat less sensitive but ensures uniformity in release characteristics across all batches produced. And one of the most important thing to understand is that QC control or quality control QC dissolution method should have a discriminatory power to detect the bad batches or mismanufactured batches or bio inequivalent batches. So quality control dissolution method should also be discriminatory towards identifying the mismanufactured batches, bio inequivalent batches, and the bad batches. So always understand that quality control dissolution method is to be discriminatory and it must have discriminatory power. So this is regarding the difference between the discriminatory dissolution and quality control dissolution testing. You can make discriminatory dissolution method as a routine quality control testing also. Also, quality control testing or quality control dissolution method should have discriminatory power. But QC testing is used at plant level to release the batch into the market, while discriminatory method is developed to have understanding about the formulation variations, and sometimes discriminatory dissolution method can also be used as a quality control dissolution test. Discriminatory power should be there for the quality control dissolution test. Also understand that quality control dissolution parameters are finalized and acceptance criteria for the QC testing is finalized based on to the bioequivalence batches and clinical batches. So this is the small video regarding difference for discriminatory dissolution and quality control dissolution. Thank you for watching the video, and I hope you have got good idea about this topic. Thank you.